In this webinar, I will briefly introduce to you the MTBseq pipeline, an automated pipeline for mapping, variant calling, and detection of resistance, mediating, and phylogenetic variants in Mycobacterium tuberculosis complex. MTBseq is an automated and modular bioinformatic pipeline for analysis of next-generation sequencing data of Mycobacterium tuberculosis samples by performing reference mapping, variant detection, annotation of variants for resistant association, and phylogenetic information, and also comparative analysis of a set of samples. This pipeline is executed only from the command line. So MTBseq is not a web tool or software that you can easily use it through a desktop computer or from an internet interface. But you have to install it on command line and use it by commands. So the pipeline consists of open source software for read mapping like SAMtools and BWA, for base calibration and refinement, MTBseq use PCART and GATK or Genome Analysis Toolkit, and for variant calling, MTBseq applies SAMtools. With the full workflow and additional functionality implemented in Perl. The modular architecture of MTBseq allows the pipeline to be fully customizable, including swapping out or adding entire complete functional module. Per default, the pipeline uses the genome of Mycobacterium tuberculosis H37RV as a basis for reference mapping, variant calling, annotation. However, any bacterial reference genome can be set by the user if the appropriate sequence and annotation files are provided. For the complete instruction of installation, description, and the usage examples of the MTBseq, please read the GitHub page of the MTBseq. So the MTBseq pipeline consists of two workflows with 7 modules encompassing a sample-specific analysis, as you can see here, and 3 modules for the comparative analysis of multiple datasets. The sample-specific workflow, as you can see here, the sample-specific workflow, which consists of these 7 steps, comprises the read alignment to the M tuberculosis H37RV reference genome, and alignment refinement, and the detection of single nucleotide polymorphisms, as well as insertions and deletions. Reported variants are annotated with applicable metadata, in particular the association with antibiotic resistance. In addition, MTBseq performs a phylogenetic classification using a set of informative SNPs, or single nucleotide polymorphism. Moreover, with MTBseq, a comparative analysis of multiple samples for phylogenetic analysis and cluster detection is also possible. While if you remember from the last webinar, with TB profiler, virus, or microbe, only the sample-specific analyses were possible. It means that you only had the chance to have information of the resistant associated mutations and the genotyping, but you couldn't do any comparative analysis for the cluster identification or phylogenetic analysis. This is one of the advantages of the MTBseq respect to the previously described pipelines. So let's briefly review the steps of the MTBseq. So the workflow starts with the raw FASTQ format sequences or the reads that you obtained from the sequencing machine. Within the mapping step, the BWA algorithm and the SAM tools are used for a generalized mapping procedure. So the step is called TBBWA. The output is a deduplicated and a sorted alignment file saved in a binary alignment format or BAN. As you can see here in this example, a BAN file produced by the TBBWA step of MTBseq pipeline. The next step is TB-refine, and in this step, the GATK is used for base call recalibration and realignment of the reads around insertions and deletions relative to the reference genome. In such regions, generally, we wish to see our reads mapped with one end of the read on the side of the indel and the rest mapped on the other side. However, the mapper that originally mapped these reads to the reference does not have information about the existence of an indel to use when mapping a given read. Thus, reads are mapped to such regions, but that only have a short part of the read representing the region on one side of the indel will typically not mapped properly across the indel, but instead be mapped with this end unaligned or into the indel region with many mismatches. The local realignment tool, in this case GATK, can use information from the other reads mapping to a region containing an indel, including reads that are located more centered across the indel and thus have been mapped with ends on either side of the indels. As a result, an alternative mapping as good as or better than the original can be generated. Moreover, for the base call recalibration, MTBseq employs a set of known resistance-associated variants of Mycobacterium tuberculosis that's used as the reference genome. The variant calling in MTBseq is a multi-step process that starts with the SAM tool's MPileup utility providing coverage information at single base resolution and produce the MPileup files from the refined mapping files of the previous step. After TBpile, the next step is for creating position lists from pileup files. Position list files capture the essential data from a mapping in a table format containing the nucleotide counts in mapped reads for each position and reference genome. And finally, in the last step of the variant calling, which is called TB variants, is the variant detection from the position lists. So variant calling can be run in different detection modes using options provided in MTBseq. For example, you can run this command MTBseq ---- step TB variants ---- snip vars. It means that you command to MTBseq to detect only the SNPs and not the insertions and deletions. Here in this slide, you can see the core metrics of the MTBseq for the variant calling and for the phylogenetic comparisons. With default setting, variants need to be indicated by 4 reads mapped in each forward and reverse orientation, respectively. At 75% of allele frequency and by at least 4 calls with a Phred score more than 20. And you know what is the meaning of the Phred score more than 20 if you remember the previous webinars, I think from the day 3. The Phred score 20, it means a error chance of 0.01%, 1 to 100. So it's a very good quality. So therefore, MTBseq reports reliably detected variants if they are present in about 75% or more of the bacterial population with default values. In the sample-specific report file, detected SNPs and insertions on deletions are annotated with respective metadata, including resulting amino acid change for SNPs in coding regions and association to antibiotic resistance. Moreover, in low-frequency detection mode, as you can see here, the parameter is different from the default mode. In low-frequency detection mode, MTBseq will consider the majority allele other than wild-type per position. This mode should preferably be used for detection of resistant-associated variants, especially if resistant subpopulations are suspected. So this is a very good detection mode. For example, consider that we have a patient under treatment, for example, for bedaquiline-containing regimen, and you get culture positives at different time points, for example, at month 3, month 6 of the treatment, and you want to see if you have any subpopulations that became resistant to bedaquiline or other drugs. So by doing whole genome sequencing and variant calling with the low frequency mode, you can detect even the mutations at 5% of allele frequency. It means that you can detect the mutations if they are present only at 5% of the bacterial population. This is very important. Moreover, we have some filters in MTBseq for the comparative analysis. In general, for doing the comparative analysis that we will review in the next slides, the repetitive regions, the PE, PPE regions of the Mycobacterium tuberculosis genome will be excluded. The resistance-associated genes will be excluded because the mutation rate in resistance-associated genes are higher respect to the other parts of the genome. And also, if two SNPs occur less than 12 base pair from each other, one of them will be excluded. So in the 6th step of the sample-specific module of MTBseq is TBstat. TBstat creates a descriptive statistic reports of the mapping and variant detection steps, giving a clear indications of overall dataset performance. For example, you can see here, for example, a FASTQ file that has been analyzed with MTBseq. You can see the information of the total reads and the total mapped reads, and for example, the percentage of the mapped reads. And these are the size of the reference genome, that is 837RV, and the GC content of the reference genome. And here you can see the total bases that were mapped to the reference genome and the GC content of this sample. So you can see that 96% we have mapped reads, and the GC content also is similar to our reference genome. So as I presented to you in previous webinars, the GC content is an important indication to see if you really sequenced the bacterial species that you wish to sequence, or you have some contamination. In this case, from the GC content also, we can understand that it can be a mycobacterium because it has a high GC content. And here you can see also the mean coverage of 17. That is not a good coverage. So this sample should be resequenced. As you remember from the previous webinars, the mean coverage of 50 is very good, and from 30 to 40 is acceptable, and from 20 to 30 is bad, and from below the 20 is ugly. So you cannot use it for the further analysis, and also the variants that have been found in this sample may not be reliable. So the recommendation is to repeat the sequencing of this sample. So by TBstats, you can have this information that you can decide for the future, for example, works that you should do for this specific sample. And finally, in the last sample-specific module in MTBseq, enables the phylogenetic classification of the input sample according to the phylogenetically informative SNPs from the literature. Again, you can see the same sample here, that after the classification based on the phylogenetically informative SNPs that taken from the literature, for example, this paper from Cole, from Homolka, and from Merker, this classification made by MTBseq, you can see that this sample that we had the low coverage, but it was for the MTBseq, it was possible to classify it. So is a Mycobacterium africanum. So it can be from lineage 5 or lineage 6, but you can see the lineage number is lineage 5. And the homolocal lineage name is West African 1b. So the full information of the lineage can be accessed by MTBseq. And based on the Cole lineage classification, the sample is belong to the lineage 5 and means West African 1. In MTBseq, a comparative analysis of multiple samples is provided by the TBjoin, TBamend modules, which can be executed for any set of samples already processed by the sample-specific workflow. It means that you have to do the sample-specific workflow for all of your samples, and then you can run the comparative analysis. So it is necessary to have the information of the sample-specific analysis in order to be able to launch the comparative analysis. The comparative analysis starts with TBjoin, that in this step, TBjoin creates a list containing information of all positions for which a variant had been detected in any of the input samples. The primary result is a list containing information of all positions for which a variant had been detected in any of the input samples. To facilitate phylogenetic analysis, variant subsets are automatically generated, filtered as you remember, for the repetitive regions, and resistant associated genes, the kind of variant, insertions and deletions also will be excluded, and the presence of other variants within a window of 12 SNPs with the same data set. So, in addition, TB amends will create a concatenated FASTA files containing all the SNPs that passed these filters, that it means that the SNPs were not present in the repetitive region, were not in the resistant associated genes, were not in insertions and deletions, and were not in presence with the other SNPs in a window of 12 base pair. So after this filtration, it will generate for you a FASTA files that includes all the SNPs that you have in your dataset. These FASTA files can be the input for the phylogenetic analysis, for example, using the RoxML, or for the minimum spanning trees that you want to calculate. And in the last step, or TB group, MTBseq does a cluster analysis based on the threshold that you define. For example, based on the threshold of 5 SNPs, MTBseq can group the samples for you. So if you see the samples in the same group, it means that the SNP difference between the members of the group is less than 5. In addition, TB groups step calculate a distance matrix of the input samples that can be also informative for you. As an example, here you can see a dataset including 18 samples that have been analyzed by MTBseq for the comparative analysis. And with TB group, the samples were grouped based on the threshold of 5 SNPs. So you can see here we have 6 group 1, 2, 3, 4, 5, and 6, and we have 2 samples that they are ungrouped. How can we interpret these? So here we can interpret that in this data set of 18 samples, we have 6 clusters that members of each clusters, for example, group 1 that consists of 4 samples, the members of the group 1, they have less than 5 SNPs distance from each other. And here you can see the distance matrix of these samples. And you can see, for example, between these and these isolates that are from the group 2, from the group 1, yeah, this 6218, and with another sample in the group 1, you can see they have 5 SNPs difference. So, and these are 61 and 65, these 2 samples are ungrouped. So it means that they have more than 5 SNPs difference from each of these groups and from themselves. So this is very important and very good feature of the MTBseq for the comparative analysis before calculation of the phylogenetic trees. At least you will have some clues of the clustering of your samples in the dataset. That is very important for the epidemiological studies. And here you can see the recent application of the MTBseq in different studies in our center that we used routinely MTBseq for the research and for the diagnosis purpose. Moreover, you can use MTBseq for other bacteria like non-tuberculous mycobacteria to do the analysis, to do the cluster analysis, the phylogenetic analysis. As you remember from the first slides, I have explained that any bacterial reference genomes can be set by the user if the appropriate sequence and annotation files are provided. So, for example, in this study, I have created a reference genome for Mycobacterium chimera and use it as a reference genome to analyze the other Mycobacterium chimera by MTBseq. So MTBseq is not limited only to Mycobacterium tuberculosis complex. Is it true that it's designed and developed for analysis of Mycobacterium tuberculosis, but it's not limited to Mycobacterium tuberculosis. You can use it for any type of the bacteria that you want. And is it only what you should do is to create a reference genome, for example. You can use it even for Escherichia coli, for Staphylococcus aureus, for other non-tuberculosis mycobacteria, Mycobacterium avium complex, Mycobacterium chimera, Mycobacterium marinum, and so on. So the only thing you need to do is to have a reference genome. And by this slide, I would like to thank you for your attention, and I will be present in the live session for you. If you have any question, you can ask me directly in the live sessions.